Welcome everyone to the clinical COVID series. This is a series organized by the Kenya Medical Association, Kenya Clinical Officers Association, National Nurses Association of Kenya, and the Clinical Psychologists Association. These Thursday webinars are the clinical ones. We have Tuesday psychological webinars, and today we are privileged to have with us Dr. Catherine Mutinda. Dr. Catherine Mutinda is a senior consultant in pediatrics and child health at Mbagathi Hospital. She holds an undergraduate degree in medicine and surgery from the First Faculty of Medicine, Charles University in Prague. She holds an M.Med. Pediatrics and Child Health degree from the University of Nairobi. And recently, she completed with distinction an MSc in medical genetics and genomics at the University of Glasgow, Scotland in the UK. She has taken part in implementation of high impact, low cost interventions in child health, notably kangaroo mother care, and establishment of the first breast milk bank in Kenya. She has participated and collaborated in various published operational studies in pediatrics and child health, geared towards clinical service delivery. She is my counterpart, the Secretary General of the Kenya Pediatrics Association, and has also been a medical superintendent at the Pumwani Maternity Hospital. Ladies and gentlemen, without much further ado, we want to welcome Dr. Catherine Mutinda to take us through the phylogenetics of COVID. And these have implications in the mutations that we are hearing are happening in Britain and South Africa, and may also come here. And she will tell us how these touches may be on the implications of the vaccine. Welcome, Dr. Catherine Mutinda.
Thank you very much for that kind introduction. I am glad to be home, and I'm glad to be talking about genetics, something that I've always loved. Today I'm going to talk about phylogenetics of COVID. When I was introduced and I was told about this talk, it's supposed to be geared toward clinical management of COVID. And phylogenetics sounds a little bit far, but it has everything to do with clinical work. And I'm going to show you how, and I hope during our discussion, we'll be able to improve on our management of our patients with the knowledge we gain today. So without much ado, this is my outline for my presentation. I'm going to go through the definitions. Everybody will be asking me what's phylogenetics. And then I'm going to talk about coronaviruses in general, then zooming into the COVID-19 in particular, talk about the novel strains that we are seeing around and the COVID-19 vaccine strategies, and see how that happens to disturb our clinical practice. So what is the definition? What's phylogenetics? Phylogenetics is just the study of evolutionary relationships.
Look, excuse.
Yes.
If you could increase your volume.
My volume. Yeah. Can you hear me better?
Yeah, now you can. We can hear you better now. Yeah.
Is that better?
That much better, Dr.
Still the moderator. Excuse me, the moderator. Yes. If you could allow me to introduce myself, I think I got caught up on the process.
So we will continue with the introduce at the end. That's what we do. So continue, Dr. Catherine Mutinda, with a bit more volume.
Is that better? My volume is okay now?
It's fine.
Thanks, obliged.
Thank you. So phylogenetics is basically drawing your family tree, where you draw your genes from, how you are related to each other, and what exactly a geneticist would do on the first meet to understand your genetic pool. So it's evolutionary relationships among biological entities. And this is very important, especially when it comes to viruses and viral pandemics, to understand especially the viruses that are zoonotic in nature, What is their origin? So that, for example, we knew that the bats were the ones that were holding our SARS and keeping us away from the bats could actually stop an outbreak. And understanding those zoonotic risks can help us contain a pandemic. We'll see why it did not work for us with COVID and why the phylogenetics needs to up its game. This is something that goes on every year, and we'll see that in Kenya it's currently going on, and we actually do the surveillances. So what are the things that in viral evolution takes place? Viruses evolve in two ways. They either drift or shift. In drifting, these are random mutations. And why do viruses have random mutations? Viruses have random mutations because their RNA polymerase is error prone. It reads its code and makes errors as it reads along, just like our DNA polymerase. But unlike our DNA polymerase, it lacks a proofreading mechanism. We do have a proofreading mechanism. Most viruses do not have a proofreading mechanism. But this is not true about the coronavirus. The coronavirus has a proofreading mechanism, and that has helped it not mutate much. And that was good news for us because we thought it would mutate a bit more slowly, giving us a chance to actually work on it and contain it with vaccines and everything else. However, we have noticed that it's actually changing that, and it's actually mutating a little bit faster. To keep it in context, the HIV virus mutates around 4 to 5 times a month. That in coronavirus, we'll do it once in a month. So at the end of the month, the HIV virus will have 4 or 5 mutations, while the coronavirus will have only one. Therefore, we thought the coronavirus was easier to work with than the HIV virus. So why mutate? When the virus mutates, it can either be deleterious, wipe itself out, or it can be beneficial and even neutral. And when it's beneficial, it helps the virus be more virulent, more infectious. And these are the things that genetic surveillance actually looks at for evolution rates and all that. And this genetic surveillance is the one that tells us how long a virus has been moving around in nature and what are the chances of it crossing over to, say, the humankind. The other way that a virus moves is by shifting. Now, shifting is a bit more complex because it either does that by recombination or segmental reassortment. Recombination is something you've heard about in biology, where similar places, that's a homologous crossing over. And within itself, the virus can actually cross over within itself, rearrange itself within itself, and come out with a new virus. And that's very common with the HIV. It does a lot of homologous crossing over with itself. But other viruses, like the parvoviruses, do a lot of recombination with the host. And this is a very important thing when they are making a vaccine for a parvovirus. This is important to note that coronaviruses are highly recombinogenic, both with self and with the host. So we'll have to keep that in mind as we discuss it later. Now, segmental reassortment is a very well-studied phenomena, and it's very common with the flu virus. We have seen a lot of flu viruses reassort, and this reassortment happens when 2 strains of a flu virus actually infect the same cell and then exchange genomic material. When they exchange genomic material, a new progeny comes out. And that new progeny can be the one that is responsible for a lot of flu pandemics that we have seen of late. And seasonal flu is one of the ways we see the different flu coming around. And that's because of the reassortment of the flu virus. And this is important to us to understand because it made from one host, so it changes host or it adapts to a new host. Like, for example, what the coronavirus is doing currently, and it's changing itself and adapt to the human host and make it more virulent and evade our immune system. So let's now move and look at the coronaviruses in general. What I want us to pull from this is that the coronaviruses are many, but I want us to look at the coronaviridae family and specifically the coronaviridae genus, and note that there are 4 of them. the alpha, the beta, the delta, and the gamma. The alpha and the beta, very interestingly named, are the ones that affect humans. The delta and the gamma are found in fish and in birds. So we're not going to talk about them, but this is important to know about them because there's always a chance that they can move and come to humans as well. So among the coronavirus, the alpha and the beta, we have the beta COVs that have got the SARS, the MERS, and now the SARS-CoV-2. Now, with that in mind, we need to look at the coronaviruses. What are they exactly? Coronaviruses are positive sense RNA viruses. When we say positive sense RNA viruses, we mean that the RNA in it is ready for translation. It doesn't need any other reorganization. It gets into the body and it translates its amino acids. It's ready for infection. And it is it's enveloped. So the envelope is very important to carry the organism through. The first strain of the human coronavirus, the B814, was seen in 1965. And since then, we have seen so many strains of coronaviruses. In fact, 15 to 30% of coronaviruses, flu, common colds that are with us today are actually caused by a coronavirus, a different kind of coronavirus. But there are 4 very important coronaviruses that have been studied greatly. That is the HKU1. This is the Hong Kong variant, the Netherlands variants, and 2 variants that were found in labs, the OC43 and the 229E. All of them have been responsible for mild to moderate upper respiratory tract infections. That's the common cold. But before the 2002, we studied a lot on the human COVID coronavirus 229E and the human coronavirus 0C43. After 2003, a lot of, due to the SARS-CoV pandemic of 2002-2003, we started getting involved and actually looking at the SARS-CoV viruses more closely. And by 10 years later, by 2012, when we started getting the MERS-CoV, we were a bit more knowledgeable about the COVIDs, and we were able to control the MERS-CoV very easily. However, we lost the battle, and we're still losing the battle with the COVID-19, which is SARS-CoV-2. So going back to the nomenclature again, I want us to look again and see that the alpha genus actually gave us the one that we were studying before the 2002 pandemic. After the 2002 pandemic, we started looking at this subgenus, and this subgenus has got a lot of influence from the bat family. So we started looking at the viruses within the bat, and that has been very helpful in understanding the SARS-CoV-2 that we are dealing with right now. Most coronaviruses are and zoonotic in nature. So as you look at your screen, you'll see that I have bats all over here and rodents. And these are known as their natural reservoirs. That's where they are actually being kept, and they are mutating in there, and they are changing. And there's a lot of surveillance that goes around this. However, there are times they move into an intermediary host. For example, when we talk about the MERS-CoV used the camel as an intermediary host. And using that donkey as an intermediary host made it very virulent and infectious to the human being, being part of the only 2 COVIDs that have been very virulent. As you can see, there are other 8 COVIDs, but only these 3 are the most virulent and have caused a lot of problems for us. So if we go back and see epidemiologically, the SARS-CoV that was in Guangdong, China, that used the civet cat as its intermediary, had a mortality rate of around 11%. Then we had this MERS-CoV that used the camel as its intermediary, and that was in 2012, and caused a mortality rate around 34%. Now, currently in 2019, we actually experienced the SARS-CoV-2 from Wuhan, and our intermediary host was either the horseshoe bat or the pangolini. And the mortality rate currently, a lot of numbers around 3.5% are being moved around, but the jury is still out there. Now, looking at the SARS-CoV-2, the sequence of SARS-CoV-2 has 50% similarities to the SARS-CoV of 2002 and a 80% similarity to the MERS-CoV. But its uniqueness stands in its spike protein, and we'll see why. So I'm going to zoom in into the SARS-CoV-2 and its morphology. The genome of the SARS-CoV-2 is actually 26 to 36 kilobases, very small, 6 to 11 open reading frames. Open reading frames are where the genes are ready to be translated into amino acids. Those nucleotides are well arranged and they are ready for translation, and that's where we get our amino acids. And then we have 9,680 amino acids being made, but they lack the HE gene. The HE gene is very common in most viruses, especially the flu virus. It's the one that helps it get into the body. However, for the SARS-CoV, in that area, it has the 5' and the 3' untranslated region. And that untranslated region is very important for its entering into our bodies. And it uses it and uses the spike protein to get in touch with our AC receptors. Interestingly, the way it's organized itself, the SARS-CoV, the first open translating area, is almost the 67% of its genome. And here it where it actually keeps the things that it requires for its translation, replication, and all other things like its helicases, the RNA polymerases. And then the rest of the genome is used for non-structural proteins and some structural proteins. And the structural proteins for COVID, and I think this picture has been seen a lot on the internet and Google, showing you the particle that is 70 nanometers to 90 nanometers big, beautiful crown, that's why the coronavirus name, and the structural proteins, that is the spike protein, which is the one that it chooses to fuse with AC receptors. And it's very important to note how beautiful it is. It has got 3 subunits. We'll talk about it a bit later. And then we have the membrane structural protein and the envelope structural protein. These are used for morphogenesis, assembly, and binding of the virus. And then we have the nucleocaspid protein, and then we have other accessory proteins. Now, up to there, you have noticed that there's a lot of homology between, or similarity between, the SARS-CoV and the SARS-CoV of 2002 and the SARS-CoV-2 that we are experiencing. But when you look at the 4 structural proteins, that homology is very high in the envelope, the membrane, and the nucleocaspid. But the spike protein is very different. And this spike protein is actually more similar to the spike protein that we see in COVID of the pangolini, making it very hard to say that this actually came from the bats directly to us. So there must have been a step where it used the pangolini and came to the humans. And that's why this virus is a recombinant virus, meaning it has pulled some things from the pangolini virus and has some original things from the original bat virus. And that is why it was very hard from the first time to get a containment, because we could not get its origins very clearly. And there's still the jury, some people still think the pangolini was a small step in its history, but it is showing that the pangolini was a very important step, because as we move on, you'll see that what our immune system responds to is to these proteins in the spike. Up to there, Dr. Kigondu, I feel like I've had a monologue. Do you think there's something I should say before we move on? I'm going very fast. Okay. I'm going so fast. Let me know and I'll slow down. So why is this all so important? Why am I going through this spike protein and running about the pangolini and the bats and evolution? It's because of the mutations. The mutations, remember we started by saying there is an RNA polymerase that is error-prone, and that error-prone RNA polymerase for the COVID has a proofreading mechanism. And that proofreading mechanism has helped us think that COVID is slow in its evolution. Well, it is slow in its evolution. And the first time we actually saw something different from the original COVID-19 of November, Wuhan, was actually in December. So one mutation occurred, and that was the D164G mutation that loosens up the spike protein to help it actually fuse much better with our AC2 receptors. And what happened? It just changed a nucleotide. The nucleotides are what we learned in biology: the adenosine, the cytosine, the guanine, the uracil. So in the RNA, the thymine is in the DNA, the uracil is in the RNA. So the base sequence in the original COVID was this. Then a mutation happened, a change, a mistake occurred that the proofreading mechanism did not correct. And that change was the change of this adenine here to a guanine. And that change had a translational effect of changing the amino acid that is actually coded from aspartic acid to glycine. And that opening changed the biochemical structure of the spike protein and made it more open. And by opening it, it made it more easily and likely to infect the human race. And that was the first time it was seen was in December in China. A second one was seen in the UK. And by June of 2020, all the samples that we have been collecting throughout the world have had this mutation. Clinically, this mutation is the one that is targeted by the remdesivir drug that we give COVID patients. And that is one of the ways that phylogenetics helps you. The second mutation that was seen came in the form of the change of N. N is an amino acid that is aspargine that at position 439 that changed to lysine. This mutation helped the virus escape the monoclonal antibodies associated with neutralizing antibodies that our bodies form. This mutation is also in this world. It's still in all our genomes, and we've seen it move very fast. Now, up to the end of the year last year, we were thinking it's doing one mutation at a time, and the whole clone has one mutation, and the next clone has another mutation. But that has easily changed in that by December of last year and November, actually, we started hearing rumors of a new mutation or a new clone that had more than one mutation, and that was moving very rapidly, meaning that the COVID is no longer slowly proofreading, but it's actually surviving. It's doing this to improve its infectiousness, its virulence, and the one that we are talking about in the UK is been named the 501YV1, or it's called the B1.7.1.7. And that mutation changes the aspargine amino acid at position 501 in the receptor binding domain to tyrosine. And this increases its affinity to bind with the receptors in the human cell. The second mutation that has been noted in the UK in the same clone is a 2 nucleotide deletion, the nucleotide 69 and 70, again in the receptor binding domain. And this helps it evade our immune system. Another important mutation is the P681H, which is immediately adjacent to the spike protein. The spike proteins have got two subunits, S1 and S2, and the S1 and S2 in between, they have the furin cleavage sites, and this helps it get in. And there is that mutation called the P681H. And this also helps it actually promotes its entry into the respiratory epithelium. So this clone is very virulent, and it's been seen in the UK. It was first seen in Kent in November, and by December, there was so much of it, and it formed a very large proportion of the population in the UK that the UK government decided to go again on lockdown because this variant of the virus was moving in a bit fast. At the same time, we also received some news from the South African surveillance studies that we not only had the the UK variants being seen there, but it also had another two variants. That's the K417 that was also helping, 417N that was also helping evade the immune system, and the E484K that was also improving the binding affinity of the virus to our AC inhibitors. Our AC2 receptors. So what is happening in our country? We are Kenyans. What's happening in Kenya? We are not very badly off. We have seen these two mutations. This is the last policy brief coming from Professor Ambrose Angweyu, also a pediatrician, I might add, but in research and with the Kemri Wellcome Trust and the MOH Kenya, they also do genetic surveillance. And these are the variants that we are seeing in our country. The deletion is there. We have seen two patients with a deletion, one in Kilifi, one in Mombasa. We have also seen, you remember the original D614G, the one that loosens up the spike protein. we are still seeing it, but it's a lot more here. So it's moved, it's here with us. Then we have this one that's from the UK, but only one patient in Lamu was in that with that one. We have this one, which is in the UK with a current mutation. We've seen one patient in Nairobi. And this is something to think about when we think about how we are actually treating our patient. Ransomware, which is the target for this mutation, works very well in our country because, as you can see, our genetic pool of the virus have got this mutation. So that's how you use this information clinically. So up to there, Kigondu, any comments? Because I'm going to the vaccine and the vaccine strategy.
Continue with the vaccine. We'll take all the questions at the end.
I'm moving a little bit fast, but I tend to talk fast. So no, moving to the vaccine and the vaccine strategies. In general, when someone is thinking of making a vaccine, they have to think of two main components. The antigen component. What is it that the pathogen presents to the immune system for the immune system to react? For COVID, it's the S protein. I've talked about this S protein. I think when you go home, just remember the S protein. And the S protein causes our bodies to form neutralizing antibodies, and also causes neutralizing antibodies that are actually aimed at the non-structural proteins. Remember, the structural proteins are 4, and there are many other non-structural proteins. So someone making a vaccine has to make sure they have an antigen that will prompt us to make neutralizing antibodies and also non-neutralizing antibodies that balance it off. Because if you do not do that, you might get an antibody-dependent enhancement of your disease, like could happen in the Zika and the Dengue viruses, and even in the SARS-CoV of 2002. So you have to get a balance between the neutralizing antibodies that you form by a vaccine, as well as the non-neutralizing antibodies. The second thing you have to think about is a signal. What will signify infection? It's very easy when you think about a live vaccine, because of course the live attenuated or even the live organism itself will signal infection. So the adjuvant that you use to signify infection when you are vaccinating is very important and very crucial. And a lot of people are going around this with very many strategies, including using mRNAs and RNases, RNases being the RNA of the virus to signify that there is a virus within the body. Finally, this is one of my last slides, the platforms. All the platforms that vaccine strategies are used, like the live attenuated, the recombinant viral vectored vaccines, inactivated vaccines, protein subunit vaccine, VLP, that's the virus-like particles vaccines, and the nucleic acid-based vaccines are out there. They are being tested. They have candidates, but the ones that have actually gotten into the market are the ones that are nucleic acid-based vaccines, and those are the mRNA vaccines that have made it to the market so far. I do not want to discuss those two that are in the market because I might be drawing one too many, and as it is right now, the mutations are changing the way we are thinking. So the jury is still out there about how well those vaccines are going to work, all of them in general. But the route has also to be thought about. There is currently the use of the IM route, and this requires you to have more than one jab, and that is the IM and the subcutaneous route. And then there's the one that you go into the respiratory mucosa, that's a spray into the nose, and that's something that we are looking at and hopefully we'll get into. So I have talked much, and those are just my references. There are just a few. And I'm open for discussion, and thank you for having me. I pray that I did not talk a lot of Greek and I made it a bit more clinical and you can use it. Thank you very much, Dr. Kigondu, for having me.
Thank you very much, Dr. Mutinda. I thought I knew a lot about COVID. We've been on it for the last almost 1 year, but I've discovered there is more to it. And I think with that, I can go to the Q&A section. Members who want to ask questions can put in their question at the bottom there. There's a Q&A section. And I can start with one. One who asks, David Kimani asks, what is the measure for antibody generation and protection? I guess, Dr. Mutinda, you may refer to that last vaccine slide where you may use it to answer that question. Yes, that one, the components, the antigen, non-neutralizing something, probably a clarification on that for David.
Thank you very much. I thought when you asked me to come on the stage, you asked me to say something on a new angle. And that's why I thought you've had this series for a while. And I thought you'd never look at the phylogenetics. And I'm glad I thought right. Thank you for having me. And to Dr. Kimani or David Kimani's question, COVID, when it gets into our body, causes us to form antibodies directly, specifically to the spike protein to neutralize the virus. The antibodies that we are forming are actually directed to the spike protein. However, if we do a lot of neutralizing antibodies, we do get into trouble. So we actually do also form non-neutralizing antibodies that are aimed to other non-structural proteins within the COVID virus itself. And that's why when we are thinking about a vaccine, the person making the vaccine has to create a balance between both the hormonal and the T-cell mediated immunity so that you don't get into trouble that we have seen of late where people are getting into a vaccine and this are what we are calling the enhanced respiratory disease or antibody dependent enhancement of COVID because of maybe one of the two immune systems being actually activated more than the other. So those are things that are put in place when you're creating a vaccine. So I don't know what I've answered. I don't know what measure he was looking for, but most of these things are started out in the lab in models, then they go to animal models before we get them as humans. Does that help? Dr. Kigondo, I think. Okay. Dr. Kigondo, I think, has just gotten disconnected. And I will try and go into the Q&A session. Celia, please help. Thank you.
Yes. Mm-hmm. So we have another question. When do we say the Pfizer vaccine is what percent effective? What do we mean?
I would like to not comment on the Pfizer or the Moderna vaccines. Currently, we have been looking at it. It's evolving every day. The jury is out on the effectiveness of this vaccine, given what I've just talked about, the changing virus. So I chose to just give 2 slides on vaccines. And if there are questions on vaccines, I beg not to answer. I am open to be invited maybe in the near future to talk about them once the jury is more confident and what we're going to do about those 2 vaccines, the Moderna and the Pfizer vaccine, as well as the Oxford vaccine that's also in the market.
All right, I think that is in order. We also have another question from David Kimani. He asked, we have not done any population. Have we done any population studies? Is it a true assertion then?
We have, as I said, I think Kenyans really don't trust themselves so much. This slide is actually a slide that is a policy brief 2021 January, given by our scientists here in Kenya. They are in Kilifi and Mombasa looking at the genetic pool of COVID. We are actually doing genetic surveillance on this COVID. And it's not the first time we are doing genetic surveillance on. We do genetic surveillance also on influenza, the flus that are the common cold that is running. This is something that is done by our government and our scientists. I'm proud to say this is work coming from Professor Ambrose Angoyu, my colleague who is a pediatrician and as well as a scientist. And I think it's about time we started looking at our homegrown results and accepting that we are as good. This work, I'm sure at the end of my presentation, right there at the end, number 12 there, this is the one that was uploaded January. That's the policy brief. So there is a lot of surveillance, population and genomic wise, to look at what we are doing and actually advise us as clinicians whether we should import even the different drugs that we are using, whether even the recovalescent plasma people are using is actually good to be used and allowable to be used. So I think we should look at our data. The policy brief come out every month, every 2 months, and if anything changes, something comes up and we are told. I don't know whether, Celia, that answers that question on the epidemiology, whether it's being done. Welcome back,
yeah.
Kigondo.
Yeah, Catherine, thank you very much. And of importance, we note that phylogenetic studies are actually going on in Kenya. And this part of the reason we are having this series is to actually show that we have these things in Kenya, and the studies can actually be used to guide our management. And it is good to hear that this is going on. I don't know, Cecilia, had you answered all the questions?
No, and we were at question. We are tackling David's Kimani question, so we can proceed on to the next questions. Kesho Kenyas ask, looking at the phylogenetics, it does not look like man-made. Maybe the person wanted to ask, looking at the phylogenetics, does it not look like man-made?
Yes, there was a time we thought this was a virus that happened as in one of the labs, and that it was so man-made, and therefore we lost some traction at the beginning of the pandemic. And that is why this picture is here to discuss that. At first, you can see SARS-CoV and SARS-CoV-2 are very similar. So people were going about the cat, but the cat was not there. And then we thought it looked like MERS. So we were thinking the camel, but the camel was not there. And it had this other, the spike protein, the combination of the spike protein that was not actually being seen anywhere else, and apart from the pangolins. That's why I'm saying, or giving a case for the fact that COVID SARS-2 or COVID-19 is actually a recombinant virus. It did not drift, it actually shifted and did a lot of recombination before we got it as humans. And that realization is what we are catching up with right now, that it must have shifted. And drifting, as these other ones did, is one of the mechanisms, but does not tell the whole mechanism of how COVID SARS-2 was made. So to your answer, COVID SARS-2 is zoonotic, but with a lot of recombination.
Thank you. Victor Ogoma asks, how was convalescent plasma therapy arrived at with regards to phylogenetics of COVID-19? I think that's a very good question.
The way we treated SARS-CoV-1, the one that was in 2002, the recovalescent plasma was seen as having neutralizing antibodies that were very important for SARS-CoV in 2002 and MERS-CoV. So the same similarity here is what led us to think that recovalescent plasma was also important. However, it is emerging that it's not important to all patients, and we have to look at genetically what the virus is containing before we use it. Otherwise, we end up with what we are calling the antibody-dependent enhancement, and we're also having enhancement of respiratory disease caused by this convalescent plasma. So that is why I'm glad to offer this talk, so that people, as you decide to give this to your patients, it's good to ask the bioinformatics of the genome, the virus genome that the patients are carrying, before you just go plainly and write a treatment sheet and maybe cause a bit more damage without understanding which virus you're looking at and what is genetic pool, because this virus is changing to avoid immune system. So if you give it a reconvalescence plasma and it has a mutation that is avoiding the antibodies, then you have not helped much. And reconvalescent plasma, currently we are seeing it's actually using it, recombining with it, and actually using it to infect us even more. Therefore, we are getting what we are calling enhancement respiratory disease out of using the reconvalescent plasma. So it's important to look this. It's no longer one size fits all. It's each patient is unique. Thank you.
Thank you. I've seen a question from the chat side on, I don't know how it may be related to this, but what is the progress made in having the WHO approved rapid tests availed for use in small facilities, including private setups? These will go along in escalating mass testing. I can take that. I think there's a list of approved COVID-19 test facilities, and there are processes going on at the Ministry of Health looking to see how antibody testing can be used in these COVID-19 era. I think that is ongoing, and there is a COVID task force that is looking into this. And I have seen another question here.
Simon, just to add on that, it's important for us to understand the difference between what you're testing. If you're doing an antibody test, it really does not tell you what you're dealing with, because some of us are very good with our antibodies, especially the T-cell immunity, and we might have a very mild, moderate disease. Remember, COVID is just the COVID-19 that's very virulent, but we have had other human COVs around, and they are still around. So when you say you're going to check for antibodies, you might not be getting the right thing. You are better off actually going to look for a PCR test, a DNA test that is going to give you the correct answer. Genetics has come a long way from 2000 when we did the first genome read, and currently you can have a turnaround time very fast with a PCR machine of giving you these answers. Unfortunately, with the genetic phylogenetics and the mutations, we might not be able to make a panel test that will just be like a nanopore or a gene expert that will just put it in and then we are ready to read. It's important for us to just trust the science. And as Dr. Kigondo has said, there's a group of people, there's a team of people that look into the science. Let's not try and give our opinions. It's very hard at this moment in a science that is quickly evolving to give a stand on anything. Thank you.
I think you had explained this, but Dr. Somba Kivungu, who is the co-convener of the Physician Health Program of Kenya Medical Association, asks that the initial story that came from Wuhan, China, was that zoonosis transmission of COVID was by eating bats. They wanted a phylogenetic comment.
A phylogenetic comment is that we did not eat bats. This is respiratory and droplet, and we mingled with the pangolins and the bats. And that's how this virus was able to slip through to the human. And that is why at the beginning, we said that it's important to understand phylogenetics so that we can contain and stop outbreaks. If we were very clear from the word go that it was the bats, it would have been very easy, like we did with the swine flu. And this was the one that is caused just a minor flu to humans, had really killed the pigs. It caused an enteritis that was easily passed to humans. For humans, it was a flu, but for the pig, it was death. It was very easy to contain the pigs and actually get rid of them. But with this virus, as I said, we thought at first it was bats. Then we have realized the spike protein has recombination from the pangolini. So we lost the battle at this point where we had not actually zoomed in onto the right zoonotic reservoir or intermediate hosts. But currently, there's a lot of work that shows that there's a mosaicism between these two that helped it slip over into the human race.
Thank you very much. My take is that it's very important to invest in science. Like as the table shows, you need to actually know exactly where that came from for you to be able to deal with it. Yvonne Thuo asks, how available are SARS-CoV-2 genetic studies to the patient? Should we test for each patient to inform management? And if so, what is the process? A very good question.
They are available. What is happening is more of surveillance. Every time you get your SARS-CoV swab done in the nose, that is pulled, even if it's going to Lancet, even if it's going to Cambridge, all that data belongs to the Kenyan people, and the Ministry of Health will take the same data for sequencing in their Kilifi or their reference lab, and the genomic pool of each of the sets is given. And that's why you can see we are able to tell you, or rather the policy brief that I saw 2 days ago, this is the latest policy brief, is telling you the variants that we have circulating in Kenya between June and October of 2020, meaning that this is something they have been looking at. So this is not something that is done per patient. This is something that is reported in a group. So that, for example, if we are treating and a policy brief comes out and says that we do not have this in our policy, in our population, then you have no business giving that patient remdesivir. But since you can see that 97% of our data has got that mutation, then you have a case to put when you put that on your T-shirt. I hope that answers.
Yeah, that's very clear. And I think that is evidence-based medicine. And it's good that it's being used in surveillance. Yvonne Thuo again asks that you comment on the persistent positivity of the COVID-19 PCR test, and whether this can be explained in the new term we have learned today, phylogenetically.
Persistent positivity. Remember, our COVID-19 or SARS-CoV-2 has a proofreading mechanism. It has a tendency not to make mistakes. So when we say we are going to wait for 2 to 12 days of incubation, it's because we are waiting for it to actually multiply enough for us to see it. Because of its slowness, it can take forever to clear, even with neutralizing antibodies being made against itself by the body. So some people who have got a very very good T-mediated cell immunity take a bit of a while to clear the positivity. That doesn't mean they are better off. What comes to mind right now is when we were dealing with HIV at the beginning, and we were dealing with the Majengo ladies, where they were saying they were exposed to HIV, but they were not getting infected. You remember such a story? It's because of the kind of HIV they were having and the receptors that they were not carrying. Ours, we have a group of people who, as much as the AC2 receptor, that is the one that the humans are using, the virus is using to get in, some people do not have a good AC2 receptor, loosely put, that makes the virus take its time to multiply, and therefore it's always there. Always there. You test, you find it there, but this person is asymptomatic. But it's always there. It's not getting in. Neither is the person producing and neutralizing antibodies, answering the question, why is it an antibody can be negative, yet the patient is positive for COVID? Because the body is not yet, you remember, it's not yet seen it. There is no prompt for it to start working. So a lot of genetic, the AC2 genetics versus the spike protein genetics, those two have to work so well together so that it can allow them to go in. Why we are seeing these mutations happening is because the virus is getting better at recognizing the ACE2 receptor. And that's why we are getting people who we can say after 14 days, they are going to be negative. But before we couldn't say that. Now we are seeing a virus that is becoming cleverer at its work. I don't know whether I've lost people or I've answered.
That is fantastic. An anonymous attendee is asking whether the new strain of SARS-CoV-2 virus reported in Europe from mutation or is it likely to be another shift or drift? I think it had been answered, but maybe you can just comment on it. They may have missed that. Muted. Sorry, you're muted. You're muted. Yes, yes.
Yes, I'm sorry. So the British mutation right now, the British strain that has been reported, as well as the one that we are seeing from South Africa, is a shift of the virus. I'll go back to the beginning of this presentation where I started from a mundane position where we started talking about drifts. And the drifts are random mutations of the virus. And these random mutations are being done so that they are beneficial to the virus itself, so that this virus is able to adapt to the new host and evade the host immune system. And in the UK and South Africa, this drift has been seen in these viruses. What's so unique about these two drifts that's making us more worried is that those clones are having more than one drift. It's carrying like 4 mutations in one. While we thought corona being a proofreading RNA virus will take its time to make a mistake, it seems it's not taking time. It's actually changing. And one of the reasons it's being said to be changing is the amount of treatment we are giving. The convalescent plasma is helping it make mutations like this mutation, the 417 mutation is very, very good at evading the antibody, neutralizing antibodies. So these things are happening, and they're happening a bit more rapidly. So we are thinking this is mutation aimed at making it a more fit virus. And this mutation is a drift, not a shift. Remember, shifts are recombinations. I hope that makes sense.
Kirtika Kesho Kenya asks why we already have the UK mutant that is not spreading.
We are not. Remember, this data is from June and October of 2012. That's the Kenyan data. The UK data, the one was reported in Kent, was the first one was being seen in June. So, and this, the 501 and the 2 nucleotide deletions had already been seen in Kenya between that time. That does not mean that we don't have them. We do have them. We do have them in the UK, and we do have them in Kenya. I think your question was, what was the question again?
You have answered it. I think it's a matter of time. It's a matter of time for the spread, to measure the spread.
Yes.
And Seth Matagaro asks whether is there a test to measure the viral load that can be used to inform how long one should self-isolate?
I'm not aware. Simon, help. Are you aware?
Yeah, I think the isolation are dependent on how long people think that someone is able to spread the virus. That is the determinant. Yeah. So we are coming to the tail end of our presentation, and I think a lot of people are very happy with the presentations. The presentations are available on the KMA website. We're also live on Facebook. Therefore, you can replay anything that you wanted to learn from Catherine as the knowledge evolves. At this point, I want to start bringing the session to a close. I want to give Steve Njoroge, the Deputy Secretary General of National Nurses Association, 1 minute to give a comment.
Thank you very much, Dr. Kingodu. Stephen Njoroge is the National Secretary, National Nurses Association of Kenya. This was a very insightful and very motivating talk. I think it's coming out clear. The virus has been exposed, whatever its source, and we are very optimistic that soon we'll have a vaccine. Soon we'll have the antiretrovirus to deal with this pandemic. So there's hope. We also appreciate that from Kenyan side, there's so much knowledge over the same, and it is being shared through this wonderful platform. So way forward, I think I call for more unity among the cadres in bringing this resource. These are knowledge is a major resource. And with unity, I am very certain we'll be able to escalate more so that our unity serves the purpose, which is to inform the health workers, secure them from further infections, and so that they can take their position in health as far as COVID management and other health challenges are concerned. So for me and my team from NNK, we appreciate AFLIDing, we appreciate KMA, we appreciate other professional bodies which are in this, and we are looking forward as we start the new year for more engagements for the betterment or for the benefit of all. So thank you very much. Stop there.
Thank you very much, Steve. Cecilia, do you want to say something about the call center?
About the call? Sorry?
It's the call center in relation to COVID.
Yes, thank you so much, Dr. Kigondu and Dr. Kimani. This is such a timely engagement and learning session, and especially when it comes to most of the calls that you are receiving from the healthcare workers about fears, about testing, about results. And so it is a great resource. And also we still urge the healthcare workers to continue utilizing the service. We have 2 lines for the call center. You are able to call in and ask your questions and talk to us. We'll provide the psychological support, and this year will be great for us, I believe. And let's continue keeping safe. And for all of us, thank you for attending. Over to you, Dr. Kigondu.
Finally, to bring this session to a close, I invite the Chief Executive Officer of Kenya Medical Association, Dr. Elizabeth Gitau, who is in charge of all these COVID trainings, and to give us a word and close the session.
Thank you very much, Dr. Kigondu, for the very good moderation, and Dr. Kimani for the amazing, mind-blowing presentation that we've had today. I would like to thank all the participants who have been with us since July when we started this program up to now, for all the knowledge that you have gotten, for the support. If we didn't have any participants, this particular course would not have gone on. We continue to generate a lot of exciting topics for you to be able to learn more about COVID and be at par with the global trends. So I am sure next week, Kigondu has very nice and exciting presentation to ensure that we get nice speakers for us. In case of any request for any topics to be done, feel free to either make a comment on any of our social media pages, whether NNK, write to us at info@kma.co.ke, and we'll respond to you and ensure that the topic that you want done will be able to be factored in. Thank you very much, Dr.
And with that, I think we bring the session to a close. I'm sure some people are still working, and thank you very much. Join us next Thursday from 2 to 3 for a COVID clinical series. Thank you very much, Dr. Mutinda.
Thank you.
Bye, everyone.